Hello, everyone. Thank you for attending today's webinar, Optimize Drug Development with Public Data and Predictive Models, Model-Based Meta-Analysis, presented by Certara. I'm Andrea Anderson, and I'll be moderating this webinar. Our speakers today are Jaap Mandema, PhD, Chief Innovation Officer, Certara, and Matt Zierhut, PhD, MBA, Vice President, Integrated Drug Development, Certara. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. All right, let's begin. Jaap, please go ahead.
Thanks, Andrea. This is Matt Zierhut. Appreciate the introduction and appreciate the opportunity to speak to you all today about an exciting topic. It is using publicly available data, mostly clinical trial results that have been published in the tools within the meta-analysis realm to really improve decision-making in drug development. So we'll break this up into a few sections. First, we want to give you guys all the value proposition for meta-analysis and model-based meta-analysis. And then we'll talk about how model-based meta-analysis, or Mbma, is different than other forms of meta-analysis. And we'll really dive into one particular example, but want to show you how meta-analysis and Mbma has been used to help answer real-world drug development questions and improve decision-making. So first, I want to talk about where does meta-analysis and Mdma fit into drug development. So I think we can all agree utilizing all potentially relevant available data when pursuing drug development, that can lead to better decisions. And that can lead to better decisions around drug development, around commercial, marketing, or really any other critical decisions you might have. But what are the data sources that we're using for these analyses, for these helpful analyses to help with our decisions? This is a very simplified 2x2 box to kind of chop up potential data sources into 4 categories. I'm sure we're all familiar with this top left category, the proprietary trial data. That is clinical trial data that is available to sponsors and companies that run the clinical trials with patient-level data. So the top row represents patient-level data. The bottom row represents summary data or aggregate data. The left column represents data from clinical trials. The right column represents data outside of clinical trials. So that right column really represents real-world data, all the data that have become recently, in the last 10 years or so, available to us outside of a clinical trial setting, right? Just a real-world patient setting. But I want to focus on this lower left quadrant today. This is the summary level data that is available from other clinical trials, published clinical trial results in aggregate, right? So we have the summary level aggregate data that anybody can tap into with journal articles, conference proceedings, conference presentations, and we can curate all that information into a database, which is what we have at Certara, our clinical outcomes database explorer, or Codex. So more generally, what can we do with meta-analysis in this setting, right? Again, it is incorporating this valuable external information into our decision-making process. So some of the key concepts or key decisions that we're going to inform: comparative safety and efficacy. Really, we want to understand what is that target that we need to beat, to differentiate whether it's safety or efficacy. We want to understand, is there a relationship between endpoints, maybe between early biomarkers and later clinical endpoints, or even across indications? We can bridge across indications that have similar treatments available. We can also create synthetic control arms with the externally available data. We can adjust across multiple studies for certain population characteristics or other factors that might be influencing trial-to-trial variability, and use that as a synthetic control arm. And we can also inform our drug development of, say, dose response or other characteristics of a new drug by borrowing information from other compounds or other trials. So we can leverage that existing information. And also we can optimize trial design, right? We can use these models to then simulate future trials and understand what is the trial design that gives us the best chance at making a correct decision or the best chance of success. So more specifically, how can meta-analysis and Mbma improve drug development? Well, as I mentioned, we really want to rigorously establish what this safety and efficacy target is needed for differentiation. That allows us to really truly understand what's needed to compete in the market. We want to be able to design cost-effective trials that really home in on improving our probability of making a correct decision. Whether that's a go or a no-go decision, we want to be able to make that decision early, and we want to make that correctly. We can, as I mentioned, we can link early endpoints with later endpoints to allow us to make those earlier decisions and really simulate those future trials with various designs to optimize them. Also, we can iteratively quantify what the probability of technical success is for a new compound as new data come in, both proprietary data for that compound as well as competitor data. As soon as it's available, we can update our models and then use that to get a more precise estimate of what that true probability of success is. And then in general, this process really just allows us to simplify knowledge transfer across many detailed trial networks or complicated indications with multiple populations and treatments. Having a curated database really simplifies that. So that curated database is really the key to all of this. And it's there's a few really critical steps in creating that database, which some of you may be familiar with, but others may not understand the rigor that goes into that process. So first and foremost, you have to formulate what your study objectives are in terms of how you're going to analyze these data. Perform the literature search across various websites. It's not just a PubMed search, right? We're looking all across potential, company websites, conference abstracts, anywhere, clinicaltrials.gov, anywhere you could possibly have relevant information from these clinical trial results. And then, of course, we review all these sources for their quality. We want to make sure that we're only including the highest quality information in the data sets, and then make the appropriate selections of those data sources. And then finally, we extract that data into a curated database that you can access through a software that we offer called Codex, or the Clinical Outcomes Database Explorer. And there's these procedures are really standardized, and you can learn more about them through the Prisma information that's the link is provided here to understand more details about this process. So the Clinical Outcomes Database Explorer, or Codex, is the software interface that we offer that allows people to interact with our 50 plus curated database across various indications. And today, I want to do a live demo to demonstrate kind of an exploratory analysis in rheumatoid arthritis to kind of whet your appetite about what this can do for, again, simple exploratory analyses, but really kind of get you thinking. So when you do a more complex analysis, or when a more complex analysis is necessary, you can use the model-based meta-analysis framework to achieve that. And that's what Jaap Mandema will be talking about in a few minutes. So let's move to. All right, here you can see the interface for Codex and the various databases that are available. We're going to focus in on rheumatoid arthritis. And you can see, if the font isn't too small, there's over 400 studies available in this database in rheumatoid arthritis. We're selecting only a subset, 11 studies that are placebo-controlled studies, 10 milligrams. And here you can perform a pairwise meta-analysis for tofacitinib 10 milligrams versus placebo, including the 11 studies that have made this comparison. And this forest plot shows you the various results you see across these trials. The symbol size represents the number of patients in these trials, so the sample size of those trials. But you'll notice that there is quite a bit of variability, right? There's a range of outcomes that you might see in the relative benefit of tofacitinib 10 milligrams versus placebo. And this pairwise meta-analysis is a quick automated analysis that allows you to understand overall the studies. This is the relative benefit of tofacitinib 10 versus placebo. And it also quantifies what the between trial variability, or the random effect across these trials is. And here you can see that is represented in this confidence interval. So it has the precision of the estimate, including the random effect or between trial variability. And the blue bar represents both the confidence interval, the uncertainty in the prediction of that weighted average, as well as incorporating that between trial variability. So a future trial would be expected to fall somewhere in this 95% prediction interval. Now, things we really aim to do is reduce that prediction interval by identifying factors that are a cause of this between trial variability. And I just wanted to point out that one thing that might be a factor here. Again, there's only 11 studies, but most of these studies are not Asian majority studies, but there's 2 tofacitinib versus placebo studies that are Asian majority studies, or they are conducted in majority in Asia. And you can see those have a bit larger treatment effect. So something we'll revisit, but it's pointing to that maybe that is one of the key factors that contributes to this between trial variability. Now, the next thing I want to do is show the network meta-analysis capability. And in order to do that, I'm going to include a few more drugs in this analysis. So I'm going to select adalimumab, 40 milligrams, and we're going to use baricitinib as kind of a use case here. And Jaap's going to go into a more detailed example using baricitinib as well. But here we're going to have 3 different doses of baricitinib, and we'll include placebo too. So it takes a few seconds to apply that filter, and now we're good to go. So we're going to perform this network meta-analysis for these 6 different treatments. And this is going to show us how these drugs compare to each other, or actually how they compare to placebo. And I'll show in the next stage, we'll show how they actually compare to each other, even though they may not have been compared head-to-head in any trial. But here, what you see is, let me see if I can zoom in. The baricitinib 8 milligram dose appears to be the most effective relative to placebo, whereas the baricitinib 2 milligram dose is the least effective amongst these treatments. And that suggests that, hey, maybe there's a dose response here, right? The 4 milligram performs somewhere between 2 and 8. The 8 milligram performs the best. There appears to be a dose response that maybe we can capture in a modeling exercise. But it also shows how baricitinib compares to these 2 more common drugs, adalimumab 40 milligrams and tofacitinib 10 milligrams. Now we can make the reference adalimumab to understand how all these drugs compare to an active comparator. This is essentially simulating a head-to-head trial, or not a trial, but the underlying truth of our best understanding of how all these drugs would perform in a head-to-head comparison versus adalimumab. And you can see, again, there's the similar evidence for a dose response, a necessary dose response for baricitinib. The higher doses appear to perform even better compared to adalimumab. And overall, tofacitinib and baricitinib, the highest dose, 8 milligrams, appear to have evidence of a significant benefit versus adalimumab 40 milligrams. Now, let's test if that Asian majority feature that we saw earlier in the tofacitinib data actually does make a difference, is a significant covariate. And in this case, we're going to apply this as a covariate separately for every intervention. And you can see when we do that, you have your non-Asian studies in green, the green square, the Asian studies in blue, the blue square. And for tofacitinib, there appears to be quite a large difference between the non-Asian and the Asian population, or Asian studies. The Asian majority trials do indeed have a stronger effect relative to adalimumab than the non-Asian trials. Whereas the control, placebo, versus adalimumab appears to be very similar. So suggesting adalimumab does not have this same Asian majority treatment effect, covariate on treatment effect. For baricitinib, we have less data, various doses. There seems to be some evidence that it does matter, especially for the lowest dose here. But that is something that we would definitely want to explore in a more detailed future analysis, model-based analysis, to really probe, where does baricitinib fit in this landscape? What are the optimal doses? What's the best way to design a trial to understand the likelihood of success of a drug like baricitinib? And those are exactly the questions that Jaap will be addressing in his case study that's coming up. But before we go to that case study, just wanted to wrap up a bit of the intro that we had here and talk about the differences between model-based meta-analysis and general meta-analysis, or pairwise meta-analysis and network meta-analysis, like the example I just showed you in Codex. So if it wasn't apparent already, model-based meta-analysis really is a tool that allows you to incorporate more parametric models, and those parameters help explain the effect of treatment, the effect of time, the effect of dose, and any other population characteristics or trial characteristics. And this is really focused to incorporate the pharmacological principles that drive the differences in outcomes of these trials. So that's really the goal, is to use these factors mostly based on pharmacological principles to explain the variability that we see across trials. And many of the features, it allows the simultaneous modeling of multiple endpoints. We didn't go into that example here, but Jaap will touch on that. And therefore, we can link early biomarkers with later outcomes, later clinical endpoints, and allows you to make predictions for late stage registration trials based on endpoints that are maybe not yet available, right? You have phase 2 endpoints that allow you to predict phase 3 outcomes. And again, like the network analysis that we showed, comparing versus placebo as well as adalimumab, the Mbma provides similar head-to-head comparisons that have never been those comparisons have never been conducted. So the advantage there is you can incorporate longitudinal data, dose ranging data, or any other kind of varying data across other characteristics. And ultimately, this is really a tool that allows you to have a predictive model, and an Mbma produces a predictive model that can be used for simulations and really help inform those most critical decisions in drug development. So with that, I think I will turn it over to Jaap to touch on a little more details about this RA case study.
Thanks, Matt, for the introduction and overview on model-based meta-analysis and showing that, in the application, the Codex application that sits on top of those databases of publicly available information, that we can actually already explore and do some quantitative assessment quite straightforwardly and on the fly. I want to dive into a particular case study and with a little bit more detail. And if we go to the next slide, really want to talk about a typical application of model-based meta-analysis, which is at the transition point from phase 2 to phase 3. Let's say we have some phase 2 efficacy safety data on a compound that we are developing, and now we're interested to make these critical decisions at that point in time. Do we go forward with this particular compound? If we go forward, at what particular dose do we go forward? And do we have a particular trial strategy or competitive advantage that we can highlight in that trial strategy, be it related to efficacy or safety, compared to other treatment options or standard of care? And the example I've picked is one of in rheumatoid arthritis, just to expand on the data you've seen earlier, looking at baricitinib, a JAK inhibitor. And on the figure here on this slide, you see the results from 2 phase 2 studies for this particular compound. So we're going to pick it up at the end of phase 2. We have this particular data available. 2 studies, like I said, we're seeing the dose response here on this slide. The x-axis is the dose, the y-axis is the response. For the typical endpoints used in rheumatoid arthritis, we them ACR 20 response in black, ACR 50 response in blue, and ACR 70 response in red. And what Matt showed you in his graphs before was the treatment effect on the ACR 20. But actually here, I'm going to expand that to look at all 3 of these at the same time. The observations in the study are the dots. These are the observed ACR 20, 50, and 70 response with their 95% confidence intervals, and the solid line that you see drawn through these points is actually our estimated dose-response relationship. So this data you typically have at the end of phase 2 that now brings up a lot of questions, which are sort of summarized on the right-hand side. Given the information that I've seen so far, what is actually my probability that this drug at a particular dose will have a better safety efficacy profile compared to other treatment options to which I haven't compared it yet in a head-to-head study? Is there a particular endpoint that will allow me better to differentiate the benefit of a compound? Can I maybe borrow some information from other drugs with a similar mechanism of action that allows me to, I'm at the end of phase 2, I'm still quite uncertain about my response. Can I put in some information, make some assumptions that will decrease my uncertainty by building in that model structure? If I go forward, should I run a head-to-head comparator study in phase 3? Obviously, that gives us a big advantage if we can show a significant benefit across a comparator or standard of care already in phase 3 going to market. And if I do that, should we first go for non-inferiority? Should we go superiority strategy right away? And what are some of the considerations that play out? I'm going to evaluate in this particular disease area. I'm going to do several studies in phase 3 in quite different patient populations. Patients that have experienced TNF inhibitors or that are naive to it, or that have experienced methotrexate or are naive to it. So I would like to get an understanding, can I project out how this drug will behave in all of these different patient populations that I have not necessarily studied yet for this particular compound? So that is the things looking forward. We can also, at this stage, often look backward a little bit and say, okay, we did 2 studies. Would 1 have been sufficient? These studies are done at 12 weeks. Should have looked at 4 weeks maybe to already get a good idea of the benefit of this particular treatment. I do a placebo control in these studies. Should I maybe have used a synthetic to reduce some of the patients being evaluated? Can I quickly expand now from my understanding in rheumatoid arthritis to adjacent indications such as psoriasis, Crohn's disease, or other areas for which these drugs are used? So you can see we have limited data at this point in time, critical decision point, lots of questions. And that's why we would run a more advanced analysis on the type of data. And I hope to show you in the next few slides how to look at each of these and provide an answer to all of those questions as we go along. So the first thing we do on the next slide is we did in this case a particular model-based meta-analysis of the time course of ACR 20, 50, and 70. So we looked at 3 endpoints at the same time so that we can also understand the correlation between those responses. We look at it over time as well. And the only reason I have an equation on the top right-hand panel here on this particular slide is to highlight that basically the only difference that I've done from the network meta-analysis that Matt showed, now for estimating that particular treatment effect, I have a function. I have a function f that's dependent upon the dose of the drug that's being given, the time point the evaluation is made, and a variety of covariates. Because as we can see, each of these dots actually is the outcome in a particular study. So these are not patients. This is a particular outcome in a study. We have repeated studies for a particular treatment, like say adalimumab. We see we have a lot of studies. We have 26 trials that have been shown repeated responses. So I can understand what is driving some of this variability across these studies. Is it patient characteristics, such as, or is it a trial setting, such as the Asian versus non-Asian that Matt already showed in the exploratory analysis? So the model comes in 2 ways: to carefully understand the impact of these patient population differences on the outcome, so that we can project these drugs as if they had been evaluated in similar patient populations, as well as to strengthen our understanding of it by building certain relationships such as dose response or time course into the models. Here we see actually the 6 TNFs that are available. We see the dose response for them in black, again, ACR 20, 50 in blue, and ACR 70 as the end, the most stringent response method endpoint in red. We can see actually very consistent dose response across these TNFs. No surprise there. They all act in a similar way. And the only difference between them is the dose that they've picked on this dose response relationship for their clinical dose with 40 milligrams every other week of adalimumab being slightly below plateau, whereas most of the other drugs are dosed at the plateau response. So just to highlight the importance of normalizing for this between study variability that we have in patient population, the next slide shows the impact of one covariate, C-reactive protein, which sort of can give you an understanding of the inflammatory state the patients are in at the beginning of the study, sort of highlighting the disease severity that we have. And actually, if I split up the TNFs in trials on the left-hand side that have a low CRP and trials on the right-hand side that have a higher CRP, and look at their dose-response relationship, we can see, especially if you look at the black and the blue lines, quite a significantly greater response in those studies that have a higher baseline CRP. So obviously, if I want to compare my trial results to that I had in phase 2 to any of these other studies that have been done, I need to normalize for these potential differences across the studies that impact the outcome. And this impact is quite large. For the typical ACR 20 response around the plateau effect, we have about an absolute difference of about 8% more patients achieving ACR 20 or 50. So going from 30 to about 55 in the low group, we go from about 30 to 65 in the higher group as a response magnitude. So it's quite important to adjust for these because they can be quite influential to explain between study differences. And CRP is a little bit more complicated, and this is actually shown on the left-hand bottom slide, where we show the typical baseline CRP over time that the study was conducted. And if we look at more recent studies, the typical CRP is lower compared to the early studies when these novel treatments, especially TNFs at that point in time, started to enter this therapeutic area. We've got the disease under much better control. the typical baseline starting point is lower. This happens a lot in a variety of disease areas. And with that came a drift in the treatment effect for the anti-TNFs over time. And here we can account for that simply by this change in the baseline CRP that occurred, when the patients got under better control, and it's in these more recent studies. So putting all of this together, we get on the next slide really what we cared most about. Similar as to what Matt showed you in the Codex application, where we look at a waterfall plot of the variety of different treatments that we have versus another treatment. Here I've analyzed the data. I can project out the results for any treatment, and there's a lot listed on the left-hand side here, versus any of these treatments that I would like. I can project the difference versus placebo. I can project the difference versus adalimumab 40 milligrams every other week, which is done here. And if we zoom in on our drug that we're interested in, and remember, we're just at the end of phase 2, so the 2 milligram dose of baricitinib is expected to be sort of similar to adalimumab, but still quite a bit of uncertainty, because obviously at this point in time, we only have limited data on this compound, maybe about 50 patients per treatment arm in each of these studies. If we zoom in on the 4 milligrams with this green arrow in the middle here, we see the expectation is to have about 8% more response in the ACR 50 and 20, but still quite a bit of uncertainty. I'm still overlapping with my confidence intervals versus the comparator that I may be most interested in, Humira. And at the 8 milligrams on the top here, I'm actually a push above that. I'm already quite certain that one will will have a larger response, but also potentially, obviously, larger risk of adverse events at that particular dose. So now what we can do is we have this understanding of the treatment effect. We can play out a variety of different scenarios. Say, okay, what if I'm planning a study, head-to-head study of my 2 milligram or 4 milligram dose of baricitinib versus adalimumab? What is the likely outcome going to be? So we take this probability distribution that we have here on the estimated effect, put that into a simulation engine to, on the next slide, show what the probability is that we'll achieve a certain outcome in a study. Now, these slides are colorful and maybe a little bit complex, but bear with me. What they're trying to show as a function of sample size on the x-axis, if we focus on the lower panel here on the bottom that shows baricitinib 4 milligrams versus adalimumab, and if we zoom in at the 400 patients per arm, a typical size of a phase 3 study in this area, and if we move up across the y-axis and look at the area under the line that's blue, which is my probability to be non-inferior at a margin of 10%. In this particular study, we see that we have a pretty high probability at this dose to be non-inferior, even though our estimated treatment effect was slightly better. We have a high probability of being non-inferior, about 92%. If I move down on the line and hit this green area, where I'm starting to get into the probability of being superior, statistically being superior to Humira, I see I have about a 50% chance to have an outcome that's significantly better compared to the comparator, in this case, adalimumab. Obviously, for the 2 milligram dose, that picture is a little bit different. All these probabilities are much lower. And in red, actually, I've got a good chance that I'll end up inferior. I still have got, about 20%, a little over 20% chance that I'll be inferior compared to this treatment. Obviously, these studies have been run by now. There's an example of a while ago. So I can actually compare our simulated outcome that we had here, which is shown in the histogram on the right-hand side. Here we're showing the distribution of the expected difference in ACR 20 in the phase 3 study, comparing baricitinib 4 milligrams versus Humira. And now I've put the actual trial results in there. And I have to repeat myself. Obviously, this distribution is generated based on phase 2 data alone. We've never studied relatively to adalimumab yet. Yet you can see there's quite good concordance between the observed outcome of about 8% to 10% difference on the ACR 20 and 50, and the estimated, which was about, or the predicted, about 7 to 8% difference on this particular outcome. And that trial both showed non-inferiority, which I believe was the primary comparison, followed by also showing a statistically significant better effect. You can see these lower confidence intervals in the actual trial results are all above 0. So they also showed a statistically significant benefit of that 4 milligrams versus adalimumab. So the next slide actually shows that not only for this particular comparator, we can actually project out the whole phase 3 strategy here. And I'm not going to go into detail of this slide, but basically a variety of studies have been done. We can simulate out the outcome based on a variety of different trial strategies and compare that to the actual outcomes in red. And we see overall across the phase 3 studies that have been run, our projections based on phase 2 were quite good. And obviously, we cannot always predict the future exactly, and that's not the objective either. The objective is to have a good enough understanding of all these factors that could potentially impact outcome. And here it's important to understand that these phase 3s are not just a bigger phase 2. They are different in the duration. So I'm going from shorter times to longer times. They're different into what treatments the patients had experienced, whether they had failed biologics, which I hadn't really evaluated for this drug, versus not. What the comparator, I already showed you that was, what particular background treatments are. So they're quite different from the scenarios we've seen in phase 2. Obviously, that's why we're doing phase 3, to evaluate all these different scenarios. But because a lot of the impact of some of these factors is actually disease specific, we can learn the impact of some of these aspects on the outcome from all the other drugs that have been evaluated. And that's why we're so successful in projecting out the outcomes for this study. So really understanding what is either drug class, or disease specific in how certain patient differences impact the outcome allows us to make us those projections. So the next slide now dives into a few more examples. One of the questions I had is, could I have made an additional assumption to better understand the dose response with the solid, or the solid blue line with the area was our estimated dose response relationship, just based on the phase 2 data from baricitinib alone, without making any additional assumptions. And then the question is, if I had assumed, because there was another JAK already with a lot of data available, tofacitinib, if I had assumed that the maximal response was the same, I could actually, in red, have decreased some of my uncertainty and be a little bit more certain about the potential benefit, because I've gotten rid of some of this uncertainty on the low end, especially of the potential benefit the compound may have had relatively to some other treatments. So that's key. I put in an assumption and I got something for that, naming some increased or decreased uncertainty. But that's the trade-off. You may want to be willing to make that assumption or not. And you don't have to. You can play out both scenarios. One, what happens if I make this particular assumption in the blue, which you've seen before? What if I do make this assumption? I could have done that as well. On the next slide shows looking at a particular adverse event. Obviously, efficacy is not the only thing that's there. On the left-hand panel, I took a snapshot out of Codex directly. So this is a screenshot out of, or downloaded plot out of the Codex application, where I'm now looking at the neutrophil effect. So I'm looking at the difference in neutrophils versus placebo for tofacitinib 5 and 10. So very similar to what Matt showed you for the clinical efficacy. We can also look at safety, do the same from our phase 2 data on baricitinib. And actually, if we look and if we analyze that from a dose response perspective, we may actually see something quite interesting, namely that 2 of these JAKs seem to have a higher ED50 or a lower potency with regards to their neutrophil effect relatively to the ACR if I compare them to another one, meaning that for a given ACR response, they'll have less neutrophil effect than filgotinib, in this particular example, another JAK inhibitor. So obviously I can expand what we're doing on efficacy on the safety side. The next slide, I'm going to skip with regards to time. So the next slide, please. I'm not going to talk in much detail on this, but we could have actually looked earlier. There's a very close correlation between the response at 1 month or 4 weeks relatively to later time points, both for a DAS score, continuous outcome in this particular indication, as well as the ACR20 odds ratio relatively to placebo shown here for week 4 and week 12 for baricitinib, actually. So we could have probably achieved the decision point based on a shorter study. The next slide shows one more point I really would like to highlight, and that's that of the endpoint. Obviously, not every endpoint is the same, and in rheumatoid arthritis, we have two that are used for registration. There's the ACR response that we've shown a lot of already in earlier slides, which is actually a US registration endpoint, or we have a DAS response, which is used in Europe more. So what I've done here is, again, take a snapshot directly out of the Codex application of all the trial data across all the different drugs that shows the difference versus control in ACR20. So how many more patients achieve ACR20 versus the difference from control in the DAS score. So what is the drop in mean DAS score relatively to control? And we can see a very tight correlation just looking at the raw data that's in the database. But these colored dots on the bottom jump out, and we've color-coded these by mechanism of action. So actually, these colored dots in the bottom are all IL-6 compounds. So it's seen for these particular compounds, given a certain ACR response, they have more effect on the DAS-related outcome, which could lead to a greater treatment differentiation and any DAS-related outcome for this particular mechanism of action, which was showed in a head-to-head study of sarilumab versus adalimumab, where in a large phase 3 study, the difference between DAS, low disease activity or remission on the bottom here, or in the orange bars on this bar graph, that difference is much larger than the difference in the ACR-related endpoints between sarilumab as an IL-6 compound versus adalimumab as a TNF. And one more minute on the last one before I move it over to the questions is that of synthetic controls. Obviously, in the RA area, it's pretty easy to recruit patients. That may not be a question you would ask, or even though it has been asked to us a few times. Can we reduce size by putting in, leveraging all that placebo data that we have? We have 180 studies in RA and combined psoriatic RA, and I'm showing just the DAS score here, typically over time for all the placebo. So these are just placebo studies now. In the red circles is the mean outcome in a particular trial over time. So there's lots of studies. We can estimate that relationship. We can explain, as you can see from the variation in those red dots, there's a lot of between trial variation in the absolute placebo response. We can explain some of that with covariates, but even after we've done that, we still have quite a bit of residual variability left. We can quantify that. And because of that, the impact of, if I had used it as an historic control using all this data that's available, was worth about 18 patients in a future study. So not a whole lot, something in this particular disease area you can quickly get to. So we can quantify that. What's the worth if we use that particular setting as a control setting in this area. And I want to point out this that I forgot to do when I showed you that very initial slide of the 2 phase 2 studies. So you remember there was quite a bit of variation in the absolute response that we saw for the ACR20. Same here, a lot of variation in the absolute change from baseline for placebo. But if you look at the relative effect of a particular treatment versus placebo, that's much better maintained across studies. Actually, there's very little or much smaller heterogeneity in the treatment effect. And that's why we can Bori, if we have an active controlled study or a placebo-controlled study, we can make such good comparisons across these studies because the residual heterogeneity in that treatment effect is actually quite low. So with that, I want to end it up. I have one more slide because this is a question, so we're preempting that. That often comes up a lot. How much time does it actually take to do all of this? And I'm not going to give you an absolute answer, but just show from a relative perspective, if we look at all the different tasks, so getting the data out into a database, enhancing all that data by cleaning it, by clustering it, by making the calculations that you can do for missing information that's there, and exploring it, all of that, about 85% of that time, you can access, or it's already set up in the Codex application. The data is there, it's fully augmented. You can do on the fly pairwise network meta-analysis as well as pairwise comparisons. So for some of these additional questions where we need a more model-based approach, we would have to take the data out and do those additional developments, but relatively that's much less time. So with that, I see we've got some questions come in. Let's move to the questioning and answering, hopefully, session in the end. So you can skip to the next one. Then thank you very much, everybody, for your attention.
Yes, thank you for those presentations. We will move on to the Q&A. There is still time to submit your questions using the Q&A tab to the left of your screen. We have a lot of great questions already, and we will try to get to as many as possible. For the first question, are these data publicly available, or is it thanks to collaboration with biotech pharma that had clinical trials?
For all the data that are available in the Codex databases that Matt was showing early on, it's all publicly available data. So it's all published data, either in a clinical, in a journal, an abstract from a conference proceeding, clinicaltrials.gov, UDRA-CT, a variety of the trial registries. All that is extracted and then organized in the databases with detailed information. Of course, where did the trials come from? What were the design, inclusion, exclusion criteria of these studies? What were all the treatments being used? What did the patients look like? What were the outcomes? A variety of different statistical analysis that were run. All that data is extracted from the literature. So it is all publicly available. Obviously, for particular clients, if interested, we can enhance that with proprietary data available or shared databases that they have access to, wrap it up in a similar paradigm so that you can look at that jointly, get the benefit of the public data together with proprietary data that you may have on these particular in this particular disease area.
All right, next question. I see how you used a joint efficacy model for ACR thresholds 20, 50, 70. Have you ever done a joint model combining efficacy and safety endpoints?
Matt, I guess I'll go again first, and you can talk about your experience. Yes, obviously, how we can compare multiple endpoints. We could also combine safety and efficacy, which is, you could do them separately. If you want to understand the correlation between those response measures, we could analyze them jointly so that we can see, okay, is the trends that we see, the magnitude, do we get more efficacy? Is that always parallel with more safety concerns, or is there a separation between that? So those are some of the additional questions you could answer by analyzing it jointly and get a direct estimate of that therapeutic index. So sort of what's my margin between the doses I need for certain efficacy response as well as safety outcomes. So the short answer is yes, many times.
Yeah, I mean, I'll echo that same, probably even the same example where, you really can home in on that therapeutic window. What is the dose that gives you enough efficacy but doesn't trigger an adverse event? And the joint model with where trials report both of those endpoints is actually a great way to probe that.
Right, another question. How frequently do you find your holdout validation exercises are successful, i.e., the observed trial results are sufficiently similar to predicted trial results?
We've done that in a variety of therapeutic areas. I've shown here RA, just one compound, but we could, do similar exercises for a lot of these compounds. Just take the phase 2 data and project out what that would mean for the later outcomes. And we've done that for not just the example I showed, but for other compounds in that area as well. Psoriasis, diabetes, dyslipidemia, a variety of areas. We've done that exercise. Always found pretty good concordance between the outcomes, actual observed outcomes, and simulated outcomes. But again, I would like to remind everybody, obviously, if I could predict everything exactly, I didn't have to do the study. I already know all these outcomes. It is more to drive the strategy. Okay, how can I optimize the trials that I'm going to run to answer some of these critical questions in that therapeutic area, and using the information that we have on other compounds in that area as best as we can to optimize that particular strategy. Matt, anything?
No, I mean, I think you've captured pretty much the essence of that one.
All right, another question. Referring to the probability of trial success portion, is it possible to use the simulations to identify the endpoint that gives the highest probability of trial success? And similar question for the comparator treatment.
Yes, as I showed for the ACR 20, 50, and 70, we have a model in each of these endpoints. So we could actually project out, excuse me, we could project out what the potential difference is across each of these endpoints. And since this is a proportion of patients, obviously how much room you have for the treatment effect depends a little bit on where the placebo response ends up in its magnitude. And actually, probably on ACR 50, we get the biggest absolute difference between control and active arms for those treatments that will have a greater benefit. So there may be a better endpoint because we have a bigger difference on that side to zoom into. Of course, there's fewer events, so that trades off that bigger difference in proportions on that outcome. So we're losing some precision. So we have to do the actual simulations to play out those scenarios.
All right, another question. Have you ever compared the accuracy of synthetic control arms based on RWD versus those based on aggregate clinical trial data?
Matt, do you want to go first? Do you want me to go on that particular one?
Yeah, I think that probably tees right into, I'm guessing, some of the work you've done in the COVID database. But I personally, I've done the synthetic control with the aggregate data, but I don't have much experience directly with the real world data. I will say I have been involved with some of those exercises, and it's frequently, it's frequently difficult to establish an accurate external control without having, you're not taking advantage of the randomized controlled aspect of trials, right? It's purely external. You're doing your best to adjust for all the appropriate characteristics that drive trial variability, but it's quite difficult to really nail that down like you do with the relative effect simulations that we were showing earlier.
Okay, yeah, I have not much to add to that particular component.
Right, another question. What do you do when some trials do not report any information for covariates of interest, or they don't report the SDSE of the trial outcome? Do you have to exclude those trials?
Yeah, that depends on the assumptions you're willing to make. Again, obviously, the first thing that we need to have a measure of outcome and a measure of the precision of that outcome. So if it's a continuous endpoint and the standard deviation is missing, then we have that question. Okay, am I going to remove those studies, or am I going to assume I understand where, what the typical standard deviation would be in a study that have been done, and impute the missing standard deviations. You can do the same for population characteristics. Often these things are correlated. So, age, gender, and the ethnicity or the race are typically reported in most studies, because that's what's always in our table one, as it were, for each of those studies. So we can build from there. And if I would say if more than 50% of the data is missing, I would not really look at that particular outcome. But let's say I have 80% of the studies that report a certain disease severity, and it's missing in 20%. I can probably impute that based on the correlation of that with other patient attributes. So that's how we often build a model for the underlying patient populations as well. So we can impute, we can do a single, you can do multiple imputation to get the impact of those covariates on the outcome in case they're missing. So I do not like to remove studies. But if you're not willing to make those assumptions, then you can leave them out, but you run the risk that you have not much left.
Yeah, and I mean,
More
I'll just add. Oh no, I was just going to add, if you have, you can explore the influence of these covariates with the studies that report it, and if you find that there is not a significant impact from those from that information, then it suggests maybe you don't even have to worry about the studies that don't report it. You don't have to impute it. You keep those studies in anyway. You don't want to lose studies for no reason, right? So if there is a relationship, then you have to make the decision, how do I impute the missing values? But if there isn't, then maybe you just keep it in anyway.
All right, next question. Do you have precedent for using this approach to replace enhance clinical studies for BLA, MA, and/or for market access payer value submissions?
Yeah, that's a good one.
Yes, our experience between Matt and I is mostly in the drug development setting, because that's a setting where you're limited in your information on the particular drug of development. So we have to make more assumptions to make out these projections as you go along. Once you get to market access at the end of the development period, of course, we have the information for that treatment, and we often do not need more complex models to understand the treatment differences. Maybe we still need to correct for the patient population differences. So yes, data out of the databases can be used to address those questions as well. I have been involved in a few applications, where we followed the drug all the way through the development, and at the end, the models get simpler because we have to make fewer assumptions into a comparison that could be made for those type of applications. But then again, it depends a little bit on what area you go to, because there's very specific models that are being expected for some of those submissions. If they are for NICE, they have very particular requirements of what you need to do. So what you would pick from an analysis is pretty much set. What data you can use? Yeah, you can use the databases to already have that data fully extracted and ready to go.
Yeah, and I think there is an opportunity to use meta-analysis Mbma with FDA submissions, or to enhance the BLA, NDA clinical studies. I think there's instances where it's definitely possible we could have enhanced that, but maybe didn't take advantage of that. But there are a few instances where meta-analysis was a benefit, particularly in oncology. There's a few examples with external or synthetic control arms to allow not a direct, but a better comparison between certain outcomes of a single arm, or maybe a trial where the comparator wasn't what was ultimately the most relevant. But there's example, I think, with Blincyto, a couple of other oncology compounds where this type of work has done. An external control arm through aggregate data has really enhanced that and helped with that approval process.
Yeah, thanks, Matt, for bringing that up. I skipped over the FDA or the regulatory submission. We've done many submissions that have included meta-analysis to enhance a variety of questions that would be their insight in the data that can provide. So that has been included in submissions. Yes.
All right, that was our final question. We had a lot of great questions today and couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. Thank you for attending this Fierce Pharma webinar and submitting so many great questions. I'd like to thank our speakers for participating and Certara for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.